Students, today we are going to discuss about next topic of this chapter, and that is molecular diagnosis. All of you know very well, to treat any disease, diagnosis play very important step. Okay, so conventional method include blood and urine analysis. From blood, serum analysis is there. So serum and blood, that is a blood analysis, and urine analysis. That is a mostly conventional method. But these methods are not suitable for early diagnosis. And you know very well, some diseases like cancer and AIDS and all that are required to early diagnosis of a disease to treat it early. Okay, so conventional methods are not suitable for early diagnosis of disease, and that's why here molecular diagnosis play very important role to treat the disease, okay, by diagnosing disease in the early stage. So it is possible by techniques like a recombinant DNA technology and PCR. You know what is a PCR? That is a polymerase chain reaction. And next one is the ELISA technique. Recombinant DNA technology, PCR technology and ELISA technology will diagnose disease early stage. Okay, that's how we can treat the disease at the early stage. First method of a molecular diagnosis is a PCR, and that is a polymerase chain reaction. And you know very well steps of a PCR, that is a polymerase chain reaction, as you have already learned in chapter number 11. So let me discuss more about the PCR in this chapter. So presence of a pathogen is normally suspected when we get the disease. Okay, whenever we get some symptoms. Okay, so we can get the idea. Whenever we get the disease, we cannot get the idea. Okay, but only after occurrence of symptom we get the idea that we have some problem. Okay, so presence of a pathogen is normally suspected only based on symptom. And sometimes what happen, a minor quantity of a pathogen, very less quantity of pathogen do not occur, means do not develop any kind of symptom. Okay, so we cannot judge that pathogen are entered in our body. By this time, the concentration of a pathogen is already very high in the body. Okay, that situation is out of control at that time, whenever symptoms are occurring. Okay, but we can diagnose when pathogen quantity is very less. Okay, on before it get the high in number. So however, very low concentration of bacteria or a virus can be detected by amplification of the nucleic acid by PCR. You know very well, PCR is the technology by using which we can get the nucleic acid in number or multiplication or amplification of a nucleic acid in billions of copies. So this method play a very important role here. So even with the low concentration of a bacteria or viruses, we can detect them and we can diagnose early. So what are the uses of PCR? Okay, so here you can see the list. Okay, let me discuss that. To detect HIV in suspected AIDS patient. Okay, is very less quantity or in the first stage. Okay, so detect mutations in genes in cancer patient also we can use PCR. Then third one, to identify many other genetic disorders. Okay, so know the genetical disorder, this technique is playing very important role. A single stranded DNA or RNA in this technique, what happened? A single stranded DNA or RNA tagged with the radioactive molecule, and that is called probe. Okay, and is hybridized with its complementary DNA in a clone of cells. It is followed by detection using autoradiography. Okay, it will be followed by autoradiography by using UV rays and radiolabel components. The clone having mutated gene will not appear on the photographic film. Okay, whatever the mutated gene or a defective gene will not appear on the photographic film because the probe will not have complementarity with the mutated gene, and as a result of that we can diagnose the disease. Recently, you know the pandemic of a coronaviruses, and the coronavirus, that is a novel coronavirus, can also be diagnosed by using PCR technique. Okay, so here also we are using PCR technique, that is a RT-PCR technique. So for that, you see the swab is collected and afterward that will be transferred to the laboratory, and in the laboratory PCR technique will be carried out. Okay, and as a result of that, this gene which is collected from the viruses will be amplified, and this amplified gene will be identified. Okay, so this is the technique for identification of or a diagnosis of a novel coronavirus also. Let me discuss about the technique in detail. So first of all, the sample will be collected from the nasal cavity. Okay, the nasal swab will be taken. Then you can understand this novel coronavirus is a RNA virus. So whatever content is there will be having RNA. Okay, and then what happened, this RNA content we can convert it into DNA first. Okay, so this enzyme which is useful here is a reverse transcriptase enzyme. Okay, so that process is called reverse transcription. Okay, so what happened, we have collected the swab and you are collecting the coronavirus from the nasal cavity by using this swab. Okay, and that coronavirus itself is a RNA viruses. So collected gene will be RNA, not a DNA. Okay, so this RNA must be converted into DNA first. Okay, and for that you have to use the reverse transcriptase enzyme. Okay, and that's why this process is called reverse transcription. Okay, so what is a transcription? We have already discussed that. Okay, transcription is the process in which DNA will be converted into RNA. That process is called transcription. But here what is done? RNA is converted into DNA. Okay, so if DNA is converted into RNA, then that process is called transcription. But here what is done? RNA is converted into DNA. That's why it is called reverse transcription. So here whatever the PCR technique will be followed is called RT-PCR. You may heard about that. Okay, so RT-PCR. What is the RT-PCR? That is a reverse transcription type of a PCR. Why here it is a reverse transcriptase type of PCR? Because coronavirus itself is a RNA viruses. That's why it must be converted first into the DNA. And after that whole process will be similar. Okay, denaturation, annealing, and extraction, elongation. Okay, so that all process will be similar. And finally, you see here the fragment will be analyzed. Okay, and based on that copying cycle, based on that number of copies, based on that it will be considered. It will be negative or a positive. Now let me watch one video for this particular
COVID-19
technique.
is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx, then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature. Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained. After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any remaining impurities from the membrane, leaving only the RNA bound to the silica gel. Once the sample is washed, the column is placed in a clean microcentrifuge tube, and an elution buffer is added. Then, a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column. And a purified RNA, which is free of protein, inhibitors, and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a premixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluorophore can be detected by the CCD camera, which converts the light that it captures into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it
Another
occurs in.
technique of a molecular diagnosis is ELISA. Okay, so what is the full form of ELISA? E for enzyme, okay, L for linked, I for immuno, then S sorbent, and A for assay. Okay, so that is enzyme linked immunosorbent assay. Okay, so ELISA is based on principle of antigen antibody interaction. Okay, so this is the nothing but it is the antigen antibody interaction. Nowadays you heard about that. Okay, for corona testing also, for corona diagnosis also, antigen method is there, antibody method is there, that is called rapid testing. Okay, so these rapid testing based on antigen and antibodies based on this technique, that is the ELISA technique. Okay, so in this case, infection by pathogen can be detected by the presence of antigen. Okay, you know very well infection is called as an infection particle is called as antigen. That may be protein, maybe glycoproteins or etc. Or by detecting the antibody synthesized against the pathogen. So we can detect antigen or maybe we can detect the antibody. This both technique, or whether it is antigen or antibody interaction, we can isolate that and identify that. Okay, and this technique is a rapid technique. Okay, so what happened during this, you can see here. So this is the antibody, this is the target antigen which may be bind with the antibody. This is the binding site. You know very well how antibody is working. Already you have learned in chapter number 10. Okay, so first of all, this antigen will bind on the antibody. You can see here, this is antigen binding site on which this antigen will be bind. Okay, so based on that, second antibody will, or a target analyte you can see, will be bind on the enzyme. Okay, that site is called enzyme linked. Okay, these you can see the complex of antigen and antibody with the bind with the enzyme. Okay, and finally this enzyme will convert the substrate into the product, and as a result of that some color change will occur, and result of that you can understand or diagnose the antigen or antibody. Okay, you can see here whole process. Sample will be collected, then sample will be introduced with the antibodies, and on the antibody particular out of these all substances, you can see this is particular red color substance which is the coronavirus antigen. Okay, will be bind on the antibody. Here you can see. Okay, rest other will not bind. Okay, so after that you will wash, so other molecule will be washed off, and then you have to add secondary antibody along with the enzyme. So if you are adding the enzyme, at that time some color change will be occur. You can read that color change and accordingly you can analyze that. Now let me watch one video to understand whole process.
ELISA, which stands for Enzyme-Linked Immunosorbent Assay, is a test used to determine the presence of antibodies in an individual. If an individual has been infected with something foreign to the body, like a virus, bacteria, or a parasite which contains antigens, the body will make antibodies against that specific antigen, and an ELISA test can be used to detect these antibodies. So for example, with the current coronavirus or SARS-CoV-2 pandemic, an individual who has been infected mounts an immune response and produces antibodies specific to SARS-CoV-2 for about 2 weeks, during which time they could be tested with an ELISA. While you would not be able to use this test to see if you have recently been infected, this test will be useful for individuals who are experiencing symptoms of the virus. Before we demonstrate our SARS-CoV-2 ELISA test, we will first explain how an ELISA works. An ELISA plate is covered with the antigen of interest, and in this case, the antigen would be particles from SARS-CoV-2, which could be any of the proteins on the surface of the virus. It's important to note that the plate does not contain the virus itself, but proteins which are the antigens to which specific antibodies are synthesized. A clinician collects fluid from the human body known to contain antibodies, which is usually blood. The red blood cells are removed so that you will have only the clear serum left, which contains the antibodies. The serum samples are added to the wells of the ELISA plate, and you can see that in this example, sample 1 contains antibodies specific to SARS-CoV-2, and sample 2 contains only nonspecific antibodies. The specific antibodies bind to the antigens on the ELISA plate. Then the wells are washed to remove any nonspecific antibodies. We cannot see these antibodies yet, so we need to perform a few more steps to be able to detect them. Next, we add the secondary antibodies specific to the primary anti-SARS-CoV-2 antibodies. The secondary antibodies each have an enzyme attached to them which can react with something else to result in a color change that we will be able to see. After 5 minutes, the secondary antibodies are bound to the primary antibodies, and we wash the wells again to remove any unbound secondary antibodies. You can see that there are no secondary antibodies in the well containing sample 2 because there were no anti-SARS-CoV-2 antibodies for them to bind to. Lastly, we add the substrate, which binds to the enzymes on the secondary antibodies. The enzyme and substrate undergo a reaction in which the enzyme converts the substrate into a visible colored product. Now we can see with our eyes which sample contained the antibodies specific to SARS-CoV-2, and we know that sample 1 tests positive for SARS-CoV-2 and sample 2 tests negative for the virus. Now we'll show you what an ELISA test actually looks like in the lab. ELISA plates usually have 96 wells like this one, but we're going to do our test in a partial ELISA plate with fewer wells since we don't need that many. We have 3 tubes containing serum samples with unknown antibodies, and we want to know if they contain SARS-CoV-2 antibodies. We also have a positive control solution which contains SARS-CoV-2 antibodies, a negative control solution which contains no antibodies, secondary antibodies, and a color reagent, which acts as the substrate which reacts with the enzymes on the secondary antibodies to produce that visible color change. We are going to add the positive and negative controls as well as the 3 samples, each to 3 wells of the ELISA plate. This is called performing the ELISA in triplicate to make sure our results are accurate. You can see that we skip a row of wells between the positive control, negative control, and the samples to prevent cross-contamination and make it easier to see what we're doing. We add 2 to 3 drops of each solution to the wells to make sure that the bottom of the well is coated with solution. When the samples and controls have all been added, we incubate them for 5 minutes to give the SARS-CoV-2 antibodies time to bind to the antigens on the ELISA plate. After 5 minutes of incubation, we will wash the wells with ELISA wash solution, which is a mild soap and low concentration salt solution. This gets rid of any nonspecific antibodies in the wells, leaving the SARS-CoV-2 antibodies bound to the antigens on the ELISA plate. Make sure you empty the wells into the sink, tipping the plate in the direction of the same solutions so that the different solutions do not mix with each other. The washing step is repeated 3 times to make sure all nonspecific antibodies are washed out. After washing, tap the ELISA plate face down on a paper towel to get rid of excess liquid. Next, we add 2 to 3 drops of the secondary antibodies to each well that had a control or a sample in it. As we saw in the ELISA illustration, the secondary antibodies will bind to any SARS-CoV-2 antibodies that are on the ELISA plate. When all the wells have secondary antibodies, we incubate for another 5 minutes to let them bind. After 5 minutes of incubation with the secondary antibodies, we wash the ELISA plate with the ELISA wash solution 3 times just like before to remove any unbound secondary antibodies. The final step is to add the color reagent, which will react with the enzyme that is bound to the secondary antibodies. As we saw in the ELISA illustration, this reaction will only occur in wells which contain SARS-CoV-2 antibodies. The result will be a visible blue color in the wells positive for SARS-CoV-2. You can immediately see this color in the positive control and in 2 of the samples, but we will give the wells time to develop to see a stronger color. After a few minutes, it's easy to see that the positive control, sample 2, and sample 3 test positive for SARS-CoV-2, while the negative control and sample 1
We
are
can
negative.
classify autoradiography in 3 ways, that is the southern blotting, northern blotting, and western blotting. Okay, in short it is called SB test, NB test, and WB test. Okay, so if you are targeting the molecule of DNA, then it is called southern blotting. If you are targeting RNA, you are analyzing RNA, then it is called northern blotting. And if you are detecting the protein, then it is called western blotting. This is the difference. Okay, and sample preparation is you can understand according to the required or a target. You can see DNA will be extracted over here. Here RNA isolation will be done. Here protein extraction will be done. And separation method is the same, that is a gel electrophoresis. You know very well what is a gel electrophoresis and how it will be work. Okay, and the membrane material is also same almost. Nylon, and here nitrocellulose. This is difference. Then probe will be here. You can understand this is a DNA molecule. Let's say probe will be according to the DNA. Means nucleic acid probe, that is homologous to the target. Here RNA, DNA or oligodeoxynucleotide, and here it will be primary antibodies. Okay, so probe label will be radiolabel enzyme. Here also radiolabel enzyme, and here only enzyme. No need of a radiolabeling. Okay, and detection method is X-ray film, chemiluminescence. Here also X-ray film and chemiluminescence. Okay, then here film and cool CCD, camera, LED. This is different kind of imaging methods. So this is your homework for today's lecture, and before we finish this lecture, let me summarize whole topic by using one animation video. So look at this video.
Apart from treatment of diseases, biotechnological methods are also used effectively for molecular diagnosis of diseases. Conventional methods of diagnosis, such as urine and serum analysis, detect the presence of a pathogen at a relatively late stage when pathogen concentration in the body becomes very high. Conversely, methods such as polymerase chain reaction or PCR and enzyme-linked immunosorbent assay or ELISA facilitate early diagnosis. PCR can detect a bacteria or virus even when its concentration is quite low in the body by amplifying their nucleic acid. It is commonly used these days to detect HIV in suspected AIDS patients, genetic disorders, and gene mutations in suspected cancer patients. The other method of molecular diagnosis is enzyme-linked immunosorbent assay or ELISA, which is based on the principle of antibody-antigen interaction. It can detect the pathogenic infection by the presence of antigens such as proteins and glycoproteins, or by detecting the antibodies synthesized against the pathogen. ELISA and PCR have completely modernized the methods of disease detection. Along with gene therapy and genetically engineered insulin, these biotechnological innovations have transformed the face of medicine and healthcare.
In next lecture, we will learn about transgenic animals. So till that, goodbye.